You think you know viruses, flu, cold, maybe COVID if you lived through lockdown, but there are things out there that make coronavirus look gentle. Viruses that melt organs from the inside. Viruses that sit silent in your brain for months before they kill you. Viruses where survival means watching half the people in the room die. These are not movie monsters. These are real. These are documented. And some of them are spreading right now. We are diving into the extreme types of viruses that have killed millions, that terrify epidemiologists, that have case fatality rates so high they make headlines when anyone survives. This is not about which virus killed the most people total. This is about which viruses are the most lethal when they infect you. The ones where your chances flip like a coin. The ones that rewrite what we think viruses can do to a human body. If you value documented virology and outbreak history like this, subscribe. It is the best way to support our work. Also comment from which city you are hearing this from. This script is researched and factual. Using sources from the World Health Organization's Disease Outbreak News, published on March 20th, 2024, the Centers for Disease Control and Prevention's epidemiological reports, updated November 6th, 2024, and peer-reviewed studies from Emerging Infectious Diseases Journal and the New England Journal of Medicine. These are not Wikipedia summaries. These are field reports from outbreak zones, autopsy findings, laboratory confirmations. Now back to the information. Let us start with the virus that changed everything about how we think about disease eradication, smallpox. Variola major. For 3,000 years it killed without mercy, fever, vomiting, then the rash, pustules covering every inch of skin, face, hands, inside your mouth, inside your throat, the bumps filled with fluid, then pus, then they scabbed. If you survived, the scars stayed forever, deep, pitted, disfiguring. But 30% did not survive. 3 in 10 dead. The numbers are staggering. Donald Henderson, who directed the World Health Organization's smallpox eradication program, reported that during the 20th century alone, an estimated 300 million people died from smallpox. 300 million. In the last 100 years of its existence, at least 500 million people died. That is more than both world wars combined. More than the Black Death. 5 million deaths per year on average in the mid-20th century. Cities emptied, economies collapsed. The virus had 2 forms. Variola major killed 30%, variola minor killed 1%, but variola major was the dominant strain, the killer. The last naturally occurring case of variola major was October 1975. A 3-year-old girl in Bangladesh, Rahima Banu. House guards posted 24 hours a day until she was no longer infectious. House-to-house vaccination within a 1.5-mile radius. Every house, every school, every healer within 5 miles. They offered rewards for anyone who reported a case. October 12th, 1977, Ali Mao Malin, a hospital cook in Somalia. He rode in a vehicle with 2 smallpox patients. 10 days later, fever, misdiagnosed as malaria, then chickenpox, finally confirmed as smallpox on October 30th. He survived. He was the last naturally occurring case of variola minor. Malin died in July 2013 from malaria while working on polio eradication. The virus that spared him took him decades later with a different disease. Then came 1978. Janet Parker, a medical photographer at Birmingham University Medical School in England. She worked 1 floor above a lab conducting smallpox research. The virus escaped through ventilation. She died, the last death from smallpox. May 8th, 1980. The World Health Assembly declared smallpox eradicated. The only human disease ever eradicated. Today, only 2 labs hold live virus. Centers for Disease Control in Atlanta, Vector Institute in Russia, under World Health Organization supervision, locked, guarded. The debate about destroying those last samples continues. Now move to something that still kills. Rabies. Once symptoms start, it is over. No virus has a higher case fatality rate once clinical signs appear, nearly 100%. 3 people in the United States have survived without vaccine after symptoms appeared. 3? Ever. The virus attacks your central nervous system. Hydrophobia, fear of water, your throat spasms when you try to swallow, hallucinations, aggression, confusion, paralysis, coma, death. Average time from symptom onset to death is 7 to 10 days. Rabies kills approximately 55,000 people every year globally, according to World Health Organization data from June 2025. Most deaths are in Africa and Asia. 99% of human rabies cases come from dog bites. The virus is in saliva. A bite breaks skin. Virus enters. But here's the thing. If you know you have been exposed, post-exposure prophylaxis works. a series of shots over 14 days. It stops the virus from reaching your brain. But many people do not realize they have been exposed. Bat bites especially, small, almost invisible. You might not even wake up. By the time symptoms appear, it is too late. The virus has reached your brain. You are going to die. In the United States, about 2 people die each year from rabies, but worldwide, the number is catastrophic. India reports the highest burden. Rural areas, limited access to vaccine, dog populations exploding, children playing, a bite, no treatment. Death weeks later. The World Health Organization aims to eliminate dog-mediated human rabies by 2030. Mass dog vaccination programs, education, access to post-exposure prophylaxis, but we are not there yet. The virus still kills daily. Now the hemorrhagic fevers, viruses that make you bleed from the inside. We start with Marburg, discovered 1967. Laboratory workers in Marburg and Frankfurt, Germany, Belgrade, Serbia. Simultaneous outbreaks. 31 infected, 7 died. The source was African green monkeys imported from Uganda for research. The workers handled infected tissue. The virus is a filovirus related to Ebola. Both cause hemorrhagic fever. High fever, headache, muscle pain, then bleeding from your gums, your nose, internal bleeding, organs failing, shock, death. Marburg virus has a case fatality rate that varies wildly. The first outbreak in 1967 had a 24% death rate. The 1998 to 2000 outbreak in Democratic Republic of Congo hit 83%. The 2017 outbreak in Uganda was 100%. Everyone died. Why does the rate vary so much? Virus strain, medical care, how quickly patients get treatment, early supportive care, rehydration, treating symptoms, improved survival. But there is no approved vaccine, no antiviral treatment. You treat the symptoms and hope. The virus spreads through direct contact with blood, secretions, organs, bodily fluids of infected people. Healthcare workers get infected frequently. Contaminated needles, poor infection control, burial ceremonies where people touch the body. The virus stays in semen for weeks after recovery, 7 weeks documented. One man transmitted Ebola virus to a sex partner more than 500 days after his first symptoms. The virus hides in the eye, the brain, the testes, immune-privileged sites. It can reemerge, cause relapse, kill you months later. World Health Organization rates Marburg as a risk group 4 pathogen, biosafety level 4 containment required. In the United States, it is a Category A bioterrorism agent. The fear is that someone weaponizes it, aerosolizes it, releases it in a crowded city. The death toll would be catastrophic. The reservoir is fruit bats, Pteropus species, Rosettus bats. They carry the virus. They do not get sick. Humans get infected through contact with bats or materials contaminated by bats, caves, mines. Once it jumps to humans, human-to-human transmission takes over. Outbreaks explode. Ebola, same family. Filoviridae, 5 species. Zaire Ebola virus is the deadliest. Case fatality rates up to 90% in some outbreaks. Average around 50%. The 2014 to 2016 West Africa outbreak was the largest Ebola outbreak in history. 28,600 cases, 11,325 deaths. It overwhelmed health systems. Guinea, Liberia, Sierra Leone. Healthcare workers died. Hospitals closed. Fear spread faster than the virus. Ebola starts like Marburg. Fever, fatigue, muscle pain, headache, sore throat, then vomiting, diarrhea, rash, impaired kidney and liver function, internal and external bleeding. Some patients bleed from the eyes, ears, nose, mouth. Others do not bleed externally but hemorrhage internally. Organs shut down. Shock, death. The virus replicates fast. Viral loads in blood reach astronomical levels. The virus attacks endothelial cells lining blood vessels. Vessels leak. Capillary permeability increases. Fluid escapes into tissues. Blood pressure drops. Multiple organ failure. There is now an FDA-approved vaccine for Zaire Ebola virus, Ervebo, highly effective, single dose, used in ring vaccination during outbreaks. Vaccinate everyone around a confirmed case, create a ring of immunity, stop transmission. There are also 2 approved treatments, Inmazeb and Ebanga, monoclonal antibodies. They work if given early. Survival rates improve significantly with early treatment and good supportive care. But in rural outbreak zones with limited medical infrastructure, death rates stay high. The virus spreads the same way as Marburg. Direct contact with bodily fluids, blood, vomit, feces, urine, saliva, sweat, semen, breast milk. Infected people are not contagious until symptoms appear, but once symptoms start, they are highly infectious. The virus stays in semen long after recovery. Over a year in some cases. Sexual transmission documented. The Ebola resistant strain does not cause disease in humans. It killed monkeys, found in the Philippines, in pigs. But humans exposed to it did not get sick. The other 4 species all caused severe disease. Moved to a virus that emerged in 1993 and terrified the southwestern United States, hantavirus pulmonary syndrome. May 14th, 1993. A 19-year-old marathon runner in New Mexico. Fever, myalgia, he collapsed, rushed to a hospital, severe shortness of breath, acute respiratory failure, dead within hours. His fiancée had died the same way just days earlier. Within 72 hours, medical investigators found 5 more cases. Young, healthy, dead from sudden respiratory failure. Initial tests found nothing. The Centers for Disease Control assembled a task force. By June 10th, using reverse transcription polymerase chain reaction, they identified a sequence from a new hantavirus. June 16th, same sequence found in deer mice trapped on site, Paramyscus maniculatus. The virus and its reservoir identified in 3 weeks. The outbreak was named the Four Corners outbreak. New Mexico, Arizona, Colorado, Utah, where the 4 states meet. In 1993, motion what? 17 of 33 cases in the Four Corners states resulted in death, 52% case fatality rate. Nationwide, 27 of 48 confirmed cases died, 56%. The virus was named sin nombre virus. Nameless virus. It causes hantavirus pulmonary syndrome, not hemorrhagic fever like Asian hantaviruses. This one attacks the lungs. Sudden onset, fever, muscle pain, then rapid respiratory decline, non-cardiogenic pulmonary edema. Fluid fills your lungs, not from heart failure, from capillary leak. Your blood vessels in your lungs leak plasma into air sacs. You cannot breathe. Oxygen levels plummet. Shock. Death. The mortality rate is around 36% overall in the United States with treatment, 50% in the initial outbreak. Why did the outbreak happen in 1993? El Niño. Unusually high precipitation in 1992 in the Four Corners region. More vegetation in spring 1993. The deer mouse population exploded, tenfold increase from the previous year. More mice, more human contact. Mice infested homes. Left droppings, saliva, urine. Humans inhaled aerosolized virus, got infected. The virus does not spread person to person in North America, only rodent to human. But there is no cure, no vaccine, only supportive care. Mechanical ventilation, extracorporeal membrane oxygenation in severe cases. If you survive the first few days, you usually recover. But that first week is brutal. Navajo oral history recorded illnesses associated with wet periods. Abundant pinyon nuts, high mouse populations. Outbreaks in 1918 and 1933. The disease was not new. It was just newly recognized by Western medicine. Early media coverage was sensational. Linked the virus to the Navajo people, promoted discrimination. The virus is named after geography, not people, but the damage was done. Since 1993, it's fewer than 800 people in the United States have been infected with hantavirus pulmonary syndrome. About 260 have died. Rare, but deadly. Malaysia. September 1998. Nipah virus. Pig farmers in Perak State started getting sick. Fever, headache, vomiting, encephalitis, brain inflammation, seizures, coma. Initial diagnosis was Japanese encephalitis, but tests did not match. Pigs were dying too. Respiratory illness. Encephalitis. Barking cough. By May 1999, 265 human cases, 105 deaths, 40% case fatality rate. The government thought it was Japanese encephalitis, delayed response. Wrong interventions. Until a virologist at University of Malaya isolated a new virus from cerebrospinal fluid of a patient, Nipah virus, named after Kampung Sungai Nipah, the village where the sample was taken. The reservoir was fruit bats. Pteropus vampirus, Malaysian flying foxes. The bats dropped partially eaten fruit into pigsties. Pigs ate contaminated fruit, got infected. Farmers had close contact with infected pigs, got infected. The outbreak spread to Singapore. Abattoir workers processing pigs imported from Malaysia. 11 cases, 1 death. The solution was brutal. 1.1 million pigs culled, destroyed. To stop the outbreak, it worked. Malaysia has had no cases since 1999. But Nipah did not disappear. It moved to Bangladesh. The transmission changed. No pigs involved. Bats to humans directly. Date palm sap. Bangladeshi people collect sap from date palm trees at night. Bats drink from the clay pots collecting sap. Contaminate it with saliva, urine. People drink raw sap. Get infected. Bangladesh has had outbreaks almost every year since 2001. Case fatality rate in Bangladesh exceeds 70%, higher than Malaysia. Why? The virus strain is different. Nipah Bangladesh strain, it sheds more virus in respiratory secretions, oral secretions, it spreads person to person. Healthcare workers get infected. Family members get infected. Outbreaks are small, 10 to 30 cases usually, but deadly, very deadly. India has had multiple Nipah outbreaks. Siliguri in 2001, 66 cases, 49 deaths, 74% case fatality rate. 75% of patients were healthcare workers or hospital visitors, person to person. Transmission in a hospital. Nadia district in 2007, West Bengal. Kerala in 2018, 17. Suspected cases, confirmed cases had high mortality. The virus is classified by World Health Organization as a priority pathogen, potential to cause epidemics, high mortality. No vaccine, no treatment, only supportive care. Studies using ferrets as models showed Nipah Bangladesh strain has higher oral shedding than Nipah Malaysia strain. Explains why person-to-person transmission occurs in Bangladesh but did not in Malaysia. The virus targets your brain, causes acute encephalitis, fever, headache, altered consciousness, seizures, coma. Some patients develop acute respiratory distress syndrome. Breathing fails. Others develop brainstem involvement. Mortality is associated with brainstem dysfunction. Positive viral culture from cerebrospinal fluid. Severe neurological damage. Yellow fever. The name comes from jaundice. Your skin turns yellow. The virus attacks your liver. Yellow fever is ancient. Descriptions date back centuries, 17th to 19th centuries. It was transported to North America and Europe. Large outbreaks disrupted economies, decimated populations. The virus is a flavivirus. Transmitted by Aedes and Haemogogus mosquitoes. There are 3 transmission cycles: sylvatic, jungle cycle, mosquitoes infect monkeys, occasionally humans in forests. Intermediate, semi-domestic mosquitoes infect both monkeys and people. Most common in Africa. Urban, infected people introduce virus into heavily populated areas. High mosquito density, low immunity from lack of vaccination. Mosquitoes transmit person to person, large epidemics. Most yellow fever infections are mild or asymptomatic, but 15% progress to severe disease, fever returns, jaundice from liver damage, abdominal pain, bleeding, vomiting blood, black vomit, kidney failure, shock, death. Among patients with severe yellow fever who develop jaundice, hemorrhage, and kidney disease, case fatality rate is 25 to 50%. Half of severe cases die. The World Health Organization estimates 67,000 to 173,000 severe infections and 31,000 to 82,000 deaths occur each year globally, most in Africa. 27 African countries and 13 Latin American countries are classified as high risk for yellow fever outbreaks. In 2016, Angola and Democratic Republic of Congo had linked urban outbreaks. 965 confirmed cases, around 400 deaths. The epidemic exhausted global vaccine supplies, diverted resources. 11 cases exported to China. West Africa has made progress. Yellow Fever Initiative launched 2006. More than 105 million people vaccinated in mass campaigns. No outbreaks in West Africa during 2015. But in 2024, armed meat was 14.6 million dengue cases were reported globally, showing arboviral diseases are exploding. Yellow fever has a safe, effective vaccine. Single dose provides lifelong immunity, but vaccine coverage is not universal. Outbreaks still happen. In 2023 across Africa, preliminary data showed 11% case fatality rate. Burkina Faso, Cameroon, Central African Republic, Chad, Republic of Congo, Côte d'Ivoire, Democratic Republic of Congo, Guinea, Niger, Nigeria, South Sudan, Togo, Uganda. All reported cases. During 1770 to 1970, yellow fever killed more soldiers posted to the West Indies than combat, 7 times the mortality rate of Canadian garrisons. The Yellow Jack, they called it. Napoleon sent an army to Saint-Domingue to reclaim Haiti. Yellow fever killed most of them, changed history. Today, travelers to endemic regions can get vaccinated. Some countries require proof of yellow fever vaccination for entry, but unvaccinated travelers still get infected. During 1970 to 2015, 11 travelers from the United States and Europe contracted yellow fever, 6 cases from West Africa, 5 from South America, 8 died, 73% mortality. Only 1 had documented vaccination history. That person survived. From 2016 to 2018, over 37 travel-associated cases, most from Angola and Brazil outbreaks. Risk for unvaccinated travelers to West Africa for 2 weeks is 50 per 100,000 for illness, 10 per 100,000 for death. South America is lower, 5 per 100,000 for illness, 1 per 100,000 for death. But during outbreaks, risk spikes. Dengue, break-bone fever. The most common arboviral disease, flavivirus, transmitted by Aedes aegypti and Aedes albopictus mosquitoes, 4 serotypes, dengue 1, 2, 3, 4. Infection with one serotype gives lifelong immunity to that serotype. Short-term immunity to the others, 2 to 3 months. After that, you can get infected again with a different serotype. And that second infection is when things get dangerous. Severe dengue, dengue hemorrhagic fever, dengue shock syndrome. The World Health Organization reports that in 2024, over 14.6 million dengue cases and more than 12,000 dengue-related deaths were recorded. Historic high. The incidence has grown dramatically. From 505,430 cases reported in 2000 to 14.6 million in 2024. The disease is now endemic in more than 100 countries. 75% of infections are asymptomatic, but those that become symptomatic range from mild fever to life-threatening hemorrhage and shock. Severe dengue occurs in about 5% of infections. Without treatment, fatality rates exceed 20%. With proper supportive care, mortality drops to about 1%. But in some settings, dengue hemorrhagic fever still kills at rates between 3 and 10%. Children in Southeast Asia are particularly vulnerable. Dengue hemorrhagic fever is a major cause of child mortality there. The virus causes sudden high fever, severe headache, pain behind the eyes, joint and muscle pain. So intense it feels like your bones are breaking. Nausea, vomiting, rash. In severe cases, plasma leaks from blood vessels, fluid accumulates in chest and abdomen, blood pressure drops, shock, bleeding from gums, nose, underskin, petechiae, purpura, internal bleeding, organ failure, death. The secondary attack rate for dengue varies. In unvaccinated people exposed to a patient, infection rates range from 40 to 90%, depending on the serotype and setting. Highly contagious through mosquito vectors. The mosquitoes breed in standing water, urban areas, tires, flower pots, water storage containers. Climate change is expanding mosquito range. Dengue is moving into new regions. Southern United States, Iberian Peninsula, Southern Europe. Cases reported where they never existed before. There are now 2 vaccines. Dengvaxia, approved 2016, but only for people previously infected with dengue. It can cause severe disease in people never exposed. Qdenga, approved 2022, suitable for adults, adolescents, children from 4 years old, but vaccine coverage is low. Most people in endemic areas are unprotected. Treatment is supportive. Fluids, manage symptoms, hospitalize severe cases, monitor for shock. No specific antiviral. You ride it out. The virus peaks around day 4 to 7 of illness, then either you recover or you deteriorate. Critical phase, plasma leakage, shock. This is when deaths happen. Close monitoring saves lives. But in rural areas without medical facilities, people die at home, uncounted. Lassa fever, West Africa, endemic in Nigeria, Liberia, Sierra Leone, Guinea, Ghana. Discovered 1969 in Lassa, Nigeria. The virus is an arenavirus. Reservoir is the multi-mammoth rat, Mastomys natalensis. The rat is everywhere in West Africa, lives in and around houses. People get infected through contact with rat urine or feces, inhalation of aerosolized virus, eating food contaminated by rats, direct contact. Once infected, 80% of cases are asymptomatic or mild, but 20% develop severe disease, bleeding from gums, nose, gastrointestinal tract, chest pain, difficulty breathing, vomiting, facial swelling, fluid in lungs, seizures, tremors, disorientation, coma, death. Case fatality rate for Lassa fever overall is about 1% in the general population, but among hospitalized patients, it is much higher, 15 to 20%. In some outbreaks, it reaches 70% or more. Nigeria recorded over 1,187 confirmed cases in 2024 across 28 states. Case fatality rate 18.5%. In early 2025, by week 11 now is SARS, Nigeria reported 535 confirmed cases and 98 deaths. 18.3% case fatality rate. Ondo, Edo, Bauchi states account for most cases. Healthcare workers get infected frequently. Inadequate infection control. Poor personal protective equipment. 31 healthcare workers affected in 2024. 4 deaths. The virus can cause permanent hearing loss. About a quarter of survivors lose hearing. It improves in about half of those cases over 3 months, but many are left deaf. Pregnant women are especially vulnerable. If infected in the 3rd trimester, maternal mortality exceeds 80%. Fetal loss approaches 95%. The virus attacks the fetus, causes spontaneous abortion, maternal hemorrhage, death. There is a treatment, ribavirin, an antiviral. If given early, it reduces mortality, but access is limited. Diagnosis is difficult. Lassa fever looks like malaria, typhoid, other febrile illnesses. By the time it is confirmed as Lassa, it may be too late. Laboratory testing requires biosafety level 4 facilities. Few countries in West Africa have them. Samples must be sent elsewhere. Delays treatment, increases deaths. The disease has a seasonal pattern. Peaks during dry season, November to April. That is when rat populations invade homes looking for food and shelter. Poor sanitation, poor housing, gaps in walls, thatched roofs, open food storage, all increase risk. Urbanization is spreading the disease. Lassa was rural. Now it is in cities, slums, high density, rats everywhere. Cases exploding. In 2018, West Africa had a major outbreak, 3,498 infections, 171 deaths. Nigeria accounted for over half, 2,121 cases, 132 deaths. The outbreak exhausted medical response resources, Médecins Sans Frontières, World Health Organization, national governments, all overwhelmed. There is no vaccine. Research ongoing. Phase 2 trials show promising results, but approval is years away. Until then, prevention is avoiding rats, trapping, poisoning, keeping food in sealed containers, filling gaps in walls, better housing, better sanitation, but poverty makes that impossible for millions. The rat population continues to grow. Climate change alters habitats. Rats move. Virus spreads. Lassa fever is on World Health Organization's priority pathogen list. Potential to cause epidemics, high mortality, limited treatments, urgent research needed. If you value documented outbreak epidemiology like this, remember to subscribe. This is the kind of information that does not reach mainstream channels but shapes how we understand infectious disease threats. Now let us finish with the full picture. Viruses do not care about your age, your health, your location. Smallpox killed kings and peasants. Rabies kills children bitten by dogs while playing. Ebola kills healthcare workers trying to save others. Hantavirus kills young marathon runners in hours. Nipah kills pig farmers and their families. Yellow fever kills travelers who skip vaccination. Dengue kills people in their own neighborhoods from a mosquito bite. Lassa fever kills pregnant women and leaves survivors deaf. These are not theoretical. These are happening right now. Nigeria reported 928 confirmed Lassa fever cases and 162 deaths by June 30th, 2024. Bangladesh continues to see Nipah outbreaks with 70+% mortality. Yellow fever is spreading to new countries. Dengue hit record numbers in 2024, 14.6 million cases, more than 12,000 deaths. Hemorrhagic fevers keep emerging. Marburg, Ebola, variants spreading. Rabies still kills 55,000 people a year, most from dog bites that could have been prevented with post-exposure prophylaxis. The pattern is clear. Climate change expands vector ranges. Mosquitoes move north, warmer winters. Longer transmission seasons. Deforestation pushes wildlife closer to humans. Bats, rodents, primates. Virus reservoirs meet human populations. Spillover events increase. Urbanization creates dense populations. Perfect for urban transmission cycles. Poverty means poor housing, poor sanitation, rat infestations, limited access to health care, late diagnosis, high mortality. Global travel means local outbreaks become international threats in hours. A case in one country can seed an outbreak on another continent before anyone knows it happened. We have tools, vaccines for yellow fever, Ebola, rabies, antivirals for some, supportive care protocols that save lives when applied early, surveillance systems that detect outbreaks, ring vaccination that contains them, But the tools are not equally distributed. Rich countries have access. Poor countries, where these viruses are endemic, do not. Vaccine stockpiles run out during large outbreaks. Medical facilities are overwhelmed. Healthcare workers get infected and die. Fear spreads. People avoid hospitals. Die at home, unreported. The true toll is always higher than confirmed cases. Research continues. New vaccines in development, new antivirals being tested, better diagnostics, point-of-care tests that give results in minutes instead of days. But research needs funding. Clinical trials need volunteers. Approval takes years. Meanwhile, people keep dying. The World Health Organization maintains a list of priority pathogens, diseases likely to cause future epidemics. Diseases we are not prepared for. Nipah is on it. Lassa is on it. Marburg is on it. Ebola is on it. Not because they might emerge, because they are emerging. Now, the irony is that we eradicated smallpox, proved that total elimination is possible, took a virus that killed hundreds of millions and made it extinct in the wild. It required global cooperation, mass vaccination campaigns, surveillance, ring vaccination, isolation of cases, decades of sustained effort. But we did it. And then we stopped. Vaccination coverage for other diseases dropped. Measles came back, polio, nearly eradicated, persists in a few countries. Yellow fever vaccines are available but not reaching people. Rabies, post-exposure, prophylaxis is effective but not accessible. The lesson from smallpox is not that eradication is easy. It is that it is possible if we commit, if we fund it, if we treat it as a global priority. Because viruses do not respect borders, an outbreak anywhere is a threat everywhere. We learned that with COVID. A virus in Wuhan became a pandemic in weeks, millions dead, economies shattered. We were lucky. COVID had a relatively low case fatality rate, 1 to 2% with treatment. Imagine if it had been Nipah, 70%, or Marburg in a bad outbreak, 80%, or rabies, 99%. The death toll would have been apocalyptic. These extreme viruses show us what is possible, what nature is capable of. A virus that makes your organs leak until you bleed to death. A virus that sits dormant in your brain, then wakes up and kills you. A virus that turns your lungs into fluid-filled sacs in hours. A virus that causes your skin to erupt in pustules from head to toe. These are not science fiction. These are real pathogens with documented case counts, mortality rates, outbreak histories, published in peer-reviewed journals, confirmed by World Health Organization, investigated by Centers for Disease Control. And here is the thing. We only know about the viruses we have discovered. How many more are out there in reservoir species? In bats, in rodents, in primates, waiting for the right conditions to spill over into humans? Climate change, deforestation, urbanization, international travel, all increasing the probability the next pandemic might not be a coronavirus. It might be a filovirus, or a paramyxovirus, or something entirely new, something we have never seen, something we are not prepared for. The data is stark. Rabies, nearly 100% fatal once symptoms appear. Marburg in some outbreaks, 100%. Nipah in Bangladesh, 70%. Ebola Zaire strain, up to 90%. Hantavirus pulmonary syndrome, 36%. Yellow fever severe cases, up to 50%. Dengue hemorrhagic fever untreated, 20%. Lassa fever in outbreaks, up to 70%. Smallpox variola major before eradication, 30%. These are not rare flukes. These are documented outbreak case fatality rates from field investigations, hospital records, laboratory confirmations. The medical response in some of these outbreaks is heroic. Healthcare workers in Ebola treatment units wearing full bodysuits in West African heat, sweat pouring, risk of infection high. Many got infected, many died. Epidemiologists tracing contacts in Nipah outbreaks, finding everyone exposed, monitoring for symptoms, isolating cases, breaking chains of transmission. Laboratory technicians handling biosafety level 4 pathogens. One mistake. One needle stick. Infection. Possible death. They do it anyway. Because someone has to. Because without them, more people die. But heroism is not a public health strategy. We need infrastructure. We need vaccines. We need antivirals. We need rapid diagnostics. We need trained healthcare workers. We need personal protective equipment. We need isolation facilities. We need surveillance systems. We need international cooperation. We need funding. Not emergency funding when an outbreak explodes. Sustained funding for research, for preparedness, for stockpiles, for capacity building in endemic countries. Because the next outbreak is not a question of if, it is when. The viruses are out there, circulating in animal populations, mutating, evolving, Waiting. A farmer clears forest for crops, disturbs a bat colony. Virus spills over. A child plays near a rat nest, gets infected. A traveler visits an endemic area, does not get vaccinated, brings virus home. An outbreak starts. And depending on which virus it is, depending on where it happens, depending on how quickly it is detected and contained, it could be a small cluster of cases, or it could be the next pandemic. The factors that determine which way it goes are often out of our control, but the preparation is not. That is on us. Some of these viruses are expanding their range. Dengue moving into southern Europe, into southern United States, cases where they never occurred. Climate models predict Aedes mosquitoes will reach new latitudes as temperatures rise. That means dengue, yellow fever. Other arboviruses follow. Areas that never had these diseases will see outbreaks. Health systems that have no experience treating them will be overwhelmed. Populations with no immunity will be vulnerable. The first few outbreaks will be brutal. High mortality. Panic. Economic disruption. Eventually, adaptation. But the cost will be measured in lives. Lassa fever is spreading in Nigeria. Case counts increasing year over year. More states affected. More healthcare workers infected. 18% to case fatality rate. That is nearly 1 in 5 confirmed cases dying. And those are just the confirmed cases. In rural areas, people die without diagnosis. Never tested. Never counted. The true burden is likely much higher. The Nigeria Center for Disease Control activates emergency operations centers every year. Multi-partner, multi-sectoral response. But the outbreaks continue. Challenges remain. Late presentation, poor health-seeking behavior, high treatment costs, poor sanitation, poor awareness, inadequate infection prevention and control. Until those systemic issues are addressed, Lassa will keep killing. Nipah outbreaks in Bangladesh are small but deadly, 10 to 30 cases. But 70% mortality, healthcare workers infected, family members infected, person-to-person transmission. The virus spreads through respiratory secretions, saliva. Unlike most other deadly viruses that require direct contact with blood or fluids, Nipah Bangladesh can spread through close contact, breathing near an infected person. That makes it more dangerous, easier to spread, harder to control. If a more transmissible variant emerged, if it spread like measles or influenza but kept the 70% mortality, the result would be catastrophic. That scenario keeps virologists up at night. Marburg and Ebola outbreaks in Africa are unpredictable. Years of silence, then sudden emergence. A case in a remote village spreads to family, to healthcare workers, to funeral attendees. Within weeks, dozens of cases, deaths mounting. Emergency response teams mobilize, isolation units set up, contact tracing begins, ring vaccination if vaccine is available. For Marburg, no vaccine, only supportive care. For Ebola, Ervebo vaccine, but you have to get it to people fast. Before the outbreak explodes, speed is everything. Delay means more deaths. The takeaway from all of this is not to panic. Panic does not help. The takeaway is to understand these viruses exist. They kill. They are documented. The data is public. The research is peer-reviewed. The outbreaks are real. Knowing they exist, knowing how they spread, knowing the risk factors, that is how you protect yourself. Avoid rat infestations. Seal food. Trap rodents. Do not drink raw date palm sap in Bangladesh. Get vaccinated for yellow fever if traveling to endemic areas. Use mosquito repellent in dengue zones. Sleep under bed nets. If bitten by an animal, especially a bat or dog, get rabies post-exposure, prophylaxis immediately. Do not wait for symptoms. If you wait, you die. If you work in healthcare and outbreak zones, follow infection control protocols. Gloves, gown, mask, eye protection. Do not take shortcuts. Shortcuts kill. For the general population in non-endemic countries, the risk of encountering these viruses is low. But travel changes that international flights move millions of people daily. A virus in one country can reach another in hours. Screening at borders helps, but asymptomatic travelers slip through. Incubation periods mean you're not sick when you board the plane. You get sick after you arrive. By then, you have exposed dozens of people. That is how outbreaks seed in new locations. That is how epidemics start. The global health community watches these viruses closely. World Health Organization issues alerts, disease outbreak news, situation reports, case counts, mortality rates, risk assessments. Centers for Disease Control maintain surveillance, travel advisories, vaccine recommendations. Research institutions study pathogenesis, how the virus replicates, how it evades the immune system, how it kills. That knowledge informs treatment, informs vaccine development, informs outbreak response. Every outbreak teaches us something. Every death is data. That sounds cold, but it is reality. We learn from tragedy, and we apply those lessons to prevent the next one, or try to. The challenge is that outbreaks in low-income countries get less attention than outbreaks in high-income countries. Lassa fever kills hundreds in Nigeria every year. It barely makes international news. Ebola outbreak in Guinea in 2014 killed thousands. It became global news only when cases appeared in the United States and Europe. Suddenly, it was a priority. Funding flowed, research accelerated, vaccine trials fast-tracked. That double standard is deadly, because by the time high-income countries care, the outbreak is already large, already out of control. If we responded early, if we supported endemic countries year-round, not just during emergencies, we could save more lives, we could prevent outbreaks from becoming epidemics, we could stop epidemics from becoming pandemics. None of this is secret. The information is public. World Health Organization disease outbreak news, Centers for Disease, control outbreak updates, peer-reviewed journals, emerging infectious diseases, the New England Journal of Medicine, The Lancet, Nature, Science. These publish outbreak reports, epidemiological studies, clinical case series, virological analyses. The data is there. The evidence is there. What is missing is political will. What is missing is sustained funding. What is missing is global coordination. What is missing is prioritization. We wait until crisis, then we scramble, then the crisis passes, then we forget, until the next one. Smallpox is proof we can do better. We eliminated a virus that killed 500 million people in a century. It took global commitment. It took decades, but we did it. The question is whether we have the will to do it again. Yellow fever could be eradicated. We have a vaccine, single dose, lifelong immunity. We know where the virus is endemic. We know the at-risk populations. Mass vaccination campaigns could eliminate urban transmission. Sylvatic cycle would remain, monkeys and jungles. But without urban amplification, large outbreaks would not happen. Human cases would be rare, manageable. It is possible. We just have to commit. Rabies could be eliminated from dogs. Dog vaccination programs work, break the transmission cycle. Humans rarely get rabies from wildlife in areas where dog rabies is controlled. Eliminate it from dogs. You eliminate 99% of human cases. World Health Organization goal is elimination by 2030. We are not on track, but it is achievable. It requires investment, requires sustained programs, requires political commitment, requires community engagement. But it is achievable. We have the tools, we just have to use them. The extreme viruses, the ones with high case fatality rates, those are harder. Ebola, Marburg, Nipah, Lassa, they emerge from animal reservoirs. You cannot vaccinate every bat, every rodent. You cannot eliminate the reservoir, so you have to break transmission at the human level. Surveillance, rapid detection, isolation, contact tracing, treatment, supportive care. Vaccines were available, antivirals in development, better diagnostics, point-of-care tests, community education, behavior change, rodent control, improved housing, sanitation, all of it together. A comprehensive approach. One health. Human health, animal health, environmental health, all connected, all interdependent. These viruses reveal the limits of our control. We like to think we have mastered infectious disease, antibiotics, vaccines, modern medicine. But viruses remind us we have not. They adapt, they mutate, they find new hosts, they spill over, they spread. And when they are deadly enough, when they kill fast enough, they humble us. They show us we are not invincible. We are vulnerable to microscopic particles of RNA or DNA wrapped in protein, invisible, silent, lethal. They have been here longer than we have. They will be here after we are gone. Our only advantage is knowledge. We can study them, understand them, predict them, prepare for them. The question is whether we will, whether we invest the resources, whether we take the threat seriously before the next outbreak, before the next pandemic, before the next virus with a terrifyingly high case fatality rate emerges and reminds us what nature is capable of. The final number to remember: smallpox killed 300 million people in the 20th century alone. We eradicated it. Rabies kills 55,000 people a year. We could eliminate it. Yellow fever kills up to 82,000 a year. We have a vaccine. Dengue hit 14.6 million cases in 2024. It is spreading. Lassa, Nipah, Marburg, Ebola, hantavirus, all documented, all deadly, all waiting for the right conditions, all preventable if we act. The science is clear. The data is public. The tools exist. What happens next is up to us. The viruses are not going anywhere. They will keep emerging. They will keep killing. Unless we do something about it. Not tomorrow. Not when the next outbreak starts. Now, while we still can, while the lessons are fresh, while the memory of what these viruses can do is still sharp, that is the only way to stop them. That is the only way forward. Knowledge, preparation, action. Everything else is just waiting for the next disaster.